Thank you for organizing this interesting conference, and it's my first time, in that, and I feel this is a gorgeous place. And so today I'm gonna to introduce one of our is a pipeline it's a pipeline work, and so we developed it's called the MyReader. So MyReader essentially is just the medical version GTP. So with the rapid development of science and technology and the advance of the biomedical literature has been routinely generated. And there are roughly 3,000 articles deposited into the PubMed repository every 24 hours, actually. And this amount of the digital publications and greatly enriched our current understanding of this medical research, okay? And it's not only help to review the, some novel biological concept, but also help us to reconstruct some of the concepts that we have already studied, okay? And however, the great opportunity also come with a great challenge. And so to deal with such large amount of those publications, and if we just use a traditional way, and if we, for example, if we just read the articles manually, and so for example like this one here, and so we just show that the number of total publications and from the January 1st 2020 to April 30th 2020, and only in this 5 months, and so we have that much of the publications. And if we want to do it manually, and for a medical, clinical practitioner, okay, and so if he just spend 30 minutes per publication and spend 8 hours per day, and without doing anything else, and it's going to take him 39 years and to finish this amount of those publications. And so this is really time consuming. And so how to basically digest all those informations and basically use or effectively use all those informations. And so our solution is we just developed this like a MyReader. So MyReader is essentially automatic learner, and which actually mimics this like a human learning process. And for human learning process, essentially it can be categorized into the 3 categories. And the first step is we processing and organizing information. Okay, and so in this step, essentially we just for human readers, and essentially we just get the key entities. Okay, and so in this step, for the NLP approach, it's just the entity extraction and also the topic model and all those summarizing informations. Okay, and another step is actually called the knowledge summarizing step. In this knowledge summarizing step, and you essentially just summarize those informations you got or the keywords you got, and into some a knowledge graph. And based on the knowledge graph, you essentially just learn the relationship between different concepts. Okay, and the last and the most challenging part is actually the higher order thinking part, like make an inference. And in the biology, and we just generate a biological hypothesis. So it's a hypothesis generation step. Okay, so this is our MyReader, and our MyReader essentially just have a few modules, and there will be more module actually just added into this this platform. And so the first module here is the topic evolution, and so in this module, eventually we just learn that for certain concept and how this concept evolved with the time. Okay, and this essentially we just build the knowledge graph, which shows the connection or relationship between different entities or different concepts. Okay, and also here we just build this like a multi-database cross-validation step. In this step, not only the publications, but also other like database informations will be included and to cross-validate whether the information we have is actually the right information or the artifact. Okay, and finally, we also have the hypothesis generation step, which means that we want to generate some hypotheses which haven't been published yet and which haven't been investigated by the researchers currently, but will be the potential like concept that appears in the future publications. Okay, and so for this, how to use this this platform. So essentially, we just input the time frame that we want to study. So for example, this one here, and we want to study the publication starting from this time and to this time. So we input this time frame. And also we input our query. So we want to just get all the publications related to the COVID-19. Okay, and then we click submit. And so we essentially get all the publications related to COVID-19, and also we just automatically and read all those publications. And so essentially, this is a pipeline for processing this MyReader. And we have the query, and we extract all the publications, and then we classify those publications, and also get the entities. And we classify them into 3 categories: the disease category, the gene category, and the drug category. Okay? And so after we doing this step, and so we perform the three modules here. Okay, so the first module is for the topic evolution. So essentially, we just track that how those like a specific topic evolution across along with time. Okay, and so here we basically just build a knowledge graph which shows the connections between different entities. And finally, in this one here, and we want to, based on the graph that we already have, and we want to make predictions that what is a potential like discovery people can have in the future. Okay, and in this step, in this module, like we further add another module which is cross-validation across different database, which basically give us more confidence of what we discovered here. And here are some like output. So for example, in the I just show you some of the examples of this output. And the data I'm using here is still like all the publications and from January 1st, 2020 to April 30th, 2020. And all the publications related to COVID-19. So this is just a demo. And so here I show the topic evolution in the gene category. And so you can see that at the beginning, and the ACE2 is actually, the center, but not, there's also some other like a topic actually appeared here. Okay, and so it's not very focused on the ACE2. And as this time goes on and move on, and so the research more and more focus on the ACE2, the graph, and the IL-6. Okay, and so this is coherent with the fact, right? Because we know that ACE2 is basically the receptor for the COVID-19 virus, right? And so it resides in the epithelial cells, and basically is a COVID-19 receptor here. And so for this tool, actually also like important regulator to trigger this like immune response to the COVID-19. And so we can see this. And this is another example about this like topic evolution in the drug category. Okay, and the remdesivir essentially, initially just was used to treat this AIDS. And so basically, that AIDS was also caused by the virus, right? And so this actually, so COVID-19 is also virus, so people use this to treat COVID-19 at the beginning. And the interesting thing here is this hydroxychloroquine. Okay, so for hydroxychloroquine, if you look at this this plot here, okay, and the green one here at the beginning, and so the increase, the research on this drug is not that much, but suddenly after this March, and so it's just explosively exponentially increase. Okay, the reason for that is actually FDA basically proved the emergency use of this hydroxychloroquine to treat COVID-19 actually on March 28th. Okay, and so basically during that time, and actually the research on that is actually expanding. But I didn't just draw the further one because the data actually the demo data I'm using is only until this April 30th. But if you see, if you see the publication, the trend of this after April 30th, and you can see there is actually a huge drop, and on June. The reason for that is actually in June, and the FDA banned the utilization for the hydroxychloroquine and to treat COVID-19 because of the irregular heartbeats and the disease and those studies. Okay, and we also output this the topic discovery, those graph from this, using the latent dirichlet analysis model. Okay, and so here the Y is a perplexity score, and the X is basically number of the topics. And so from this one, we can see that actually for this particular data and there's 4 significant topic. Okay, and if we further study this 4 topic, and so we see this the top keyword of this 4 topic. Okay, and so the first topic here basically just cover all those radiology related entities, and so you have we have the imaging and the radiological and then more of that. Okay, and so this is a pattern for the first topic. And if you look at that, and there is a big increasing from the January 1st and to February, right? And then the increase becomes flat. The reason for that is actually, starting from the January 1st to February, and the PCR technique is still, is not available at the time. And so the only way to identify the COVID-19 at the time is actually using the radiology technique, and especially it's just the ground glass technique here. And so, that is why we see that there is a, large increase in this period, and then it's become flat. And the second actually topic is about the animal baths, and so it's basically, this related to the, virology studies for this, because from the word, and you can see that, right? So it's a virology studies for the COVID-19 in this. Basically, people just study the phylogenetic and whether it's the virus coming from the bat or coming from the poultry or others. Okay, and the third topic here actually also corresponding to the the healthcare and the educations. And so you can see that at the beginning, for this one here, and the increase is not, is pretty flat for this category. And then, however, after March, and the research on this has been greatly increased. And the reason for that is actually, during that time, and so there's already the public policy actually involved, which actually quarantine and all those all those politics has been released. Okay, and the last one is actually genetic related, right, studies, and so is the ACE, and so also like the RT-PCR approach. Okay, so in the fourth one, like after the February, and so the testing strip is already available. So after that, not so much like a study on this thing here, because FDA is already approved the use of that. And here's also like a knowledge graph. So for all the entities, and we just based on their co-occurrence, and we construct this like a weighted network. And here we also propose the enrichment analysis. And so for this enrichment analysis, and so we basically remove that the connections and which are significant but not related to COVID-19. So here in this studies and the final output graph, so only report that the connections that related to the COVID-19 studies. Okay, so for example, this one here, this two, the anxiety and the stress, and they always co-occurrent. However, they just have no relationship with the COVID-19. So in the enrichment study, and this has been removed. And here is another example. And so in this like gene network, and we see that these two things, the IL-6 and the TNF alpha, and they are heavily connected and always co-occurrence in the extracted publications. And however, using our proposed enrichment analysis, and this connection has been removed. The reason for that is actually these two things here, and they just they are the central factors in alter this like a cytokine network of the uremia. So basically, they participate in all the different like biological process, not have to be not just for the COVID-19 like reactions. And so this is why we removed that. And we also add up this like the cross-validation modules. Okay, so in this module, and we have our like literature-based network, which constructed only using the publications and related to COVID-19, right? And so we also have some other like knowledge-based networks. So for those networks, we constructed using the database like Entrance, okay, or like biobank, okay, using those like networks. And so we just cross-validated the information we obtained from this literature-based network with other knowledge-based network. And so for the item or entities or connections appeared in both of these two, and so we have more like confident that this discovery or these connections and is a true discovery, right? And we also have something like maybe only appears in the literature but not in the database, or this like this area like only in the database but not in the in the literature studies. Okay, and so this part actually may also okay, interesting part. And almost done. And so here is a cross-validation, and the final module, actually we make a prediction. Okay, so this is the network that we already have, not this, actually, we have already constructed the knowledge graph, right? And so essentially using some like a network model, and we also make a prediction. So the prediction is more like, the generating, like a ChatGPT, right? You have the input and you generate the new thing. Yeah, finish. And so here is the network that, or the, predicted link that we have. And so this predicted link, this is, the link we predict using the given data, the data before April 30th. And this is the validation. So this validation is using the publication after April 30th. Okay, and we found out that 71% of the connections we made here actually appeared in the publications that after this, April 30th. And finally, the last one here, and we also discovered this particular this in here, and this medicine originally was proposed to treat this hepatitis C. And eventually, actually, from our study, we see that it's also related to the, we predicted it's also related to COVID-19. And later on, actually, it was also released by the, FDA that this could be used to help the recovery after the COVID hospitalizations. And finally, this work is, a joint work with my previous student Hui Min, and who is currently assistant professor in Boston University. And this is Terry Ma, and he's a computer scientist in the CMU. And so all the platform and all those UI UX design and all those things, and actually was done by him. And the large, also how to get this, perform the ALP and all those studies was conducted by him. And all those, the statistical models, and was studied by her. Thank you.
Thank you. We have one short question. Yes.
Yes.
There are many